The first thing, before the actual content of today's lecture, you need to know about the different components of the lymphatic system. The lymphoid system or the lymphatic system. The cells present in the lymphatic system. I'm sending you the picture which I just took. मैंने अभी एक तस्वीर बनाई है. ये recording screen किस तरह की दिखती है from my side. Okay, WhatsApp किया मैंने आपको. So let's start formally with some information about the lymphatic system before we proceed. There are the lymphatic system which we have studied in our earlier and initial lectures. The cells which are circulating in the lymphatic system, they are called as lymphocytes. तो ये term जो है वो आपके दिमाग में confusing नहीं होना चाहिए. Lymphocytes, the cells which are circulating in the lymphatic system, and they are actually the cells which are responsible for the protection. They are actually the cells which are responsible to invigilate the different parts of the body. These are the cells which not only detect the things, but also they are responsible to kill the stuff depending on the nature of the pathogen. They are not only moving in the circulatory system, I mean in the blood, but those cells are also came outside the blood vessels and stays within the intracellular spaces, because much of the infection, they manage to penetrate the spaces between the cells. The first part of that lymphatic system is called as primary lymphoid organs. The primary lymphoid organs are the source of lymphocytes. Are the source of lymphocytes. Your lymphocytes पैदा करने की जगह, primary lymphoid organs. And you know, the one, the bone marrow, the other, thymus. They're the primary lymphoid organs. The beta lymphocytes, they mature in the bone marrow, while T lymphocytes, after they have been generated in the bone marrow, they transfer to the thymus and they mature over there. That's the little difference between them. Both of them were generated within the bone marrow, but the beta cells, they not only produce in the bone marrow, but they also get matured in the bone marrow. But on the other hand, the T cells, they were born in the bone marrow, and after that they get mature when they reach into the thymus, and that is why they're called as T lymphocytes. The other things which are associated with the lymphoid system are the lymphatic vessels, the lymphatic system, the small vessels from the intracellular spaces, lymph capillaries collect lymph from the tissues, from the intercellular spaces, merge together to make a bigger vessels. Those bigger vessels merge together to make even bigger vessels, and eventually connecting with our central nervous, with our circulatory system. All the lymph is then go back to our bloodstream, and then once again become part of our blood circulatory system. The third thing is the secondary lymphoid organs. They are adoptive immune responses in order to respond to the new things, in order to see or meet with the new stuff. There is a pathogen which can be dealt with the help of innate immune system. We can deal with it. But if that pathogen is new, our lymphatic system, okay, you don't need to put any attendance anymore, okay? That's okay. You have done. So if an adoptive immune system is required to be activated, or the adoptive immune system needs to know about the trouble, the cells of the adoptive immune system, did you remember, macrophages or antigen presenting cells, they needs to spend time with each other and with the trouble. They need to spend some time with each other and with the trouble. So that thing or that situation was created because with the help of the lymph nodes, and the spleen. So the lymphatic system contains the small, I must say, the small, क्या कहते हैं उसको, चुराहा. It's a kind of chopal. So lymphatic vessels from different parts of the body, they connect and create a big hall, like a city hall, where everybody is coming and spend some time with each other in those places. Those places are called as lymph nodes. So lymph nodes are the places where lymphocytes spend enough time with each other. And you can guess, if a macrophage is coming into the lymph node with the information about the antigen, that information can be presented to the T cells. Because in the lymph node, in the lymph nodes, they will going to spend a reasonable time with each other, so they have enough time to exchange information. So lymph node give that environment to the macrophages, which are coming with the information about the antigen, and the naive T cells to spend some time with each other so they can exchange information with each other. It's a kind of a place where different things from that part of the body, they come and they drain their lymph at one place. That little bulge or a sac-like structure is called as lymph node. The spleen is doing the same thing. You might remember, or you might heard about the lymph nodes. Lots of lymph nodes are present in the armpit regions. Lot of lymph nodes are present in the armpit regions, and then it is also present in other parts of the body as well. But especially, I mentioned about the presence of lymph nodes in the armpit region it is because when a person, Allah na kare, is diagnosed with breast cancer, its body started to recognize the tumor, if the tumor is big enough, and the adoptive immune system will try to start learning about the trouble, will start creating the weapons against that tumor cells. So lots of immune activity is going on in that region, in the breast region. The most nearest lymph nodes near the breast is the lymph nodes of the armpits. So what that means, if a tumor is present in the breast, the fighters or the immune cells which are fighting against that tumor will going to be accumulated in those lymph nodes. If a tumor or the lymphocytes, they are accumulating nearby the tumor or within the tumor, that means the bad cells are present at one place. Such tumors are benign or non-metastatic. They're not spreading. But if the doctor assumes, with the help of the conditions and the reports, that this tumor is spreading, or the doctor wants to investigate whether this tumor is spreading or not, the doctor asks the people from the medical laboratory that take some samples of the cells from the lymph nodes of armpits as well. तो कुछ lymph nodes से भी वो sample को निकालते हैं syringe की मदद से. They take some sample out of the lymph nodes as well. So if you able to see the epithelial cells, the cells of the tumor, and the immune activity in the lymph node, that can be a reason for the diagnosis of that cancer as a metastatic breast cancer. So these things are important and related. It can be considered as, because the tumor is now trying to spread out of the place, the first time the tumor will going to see the lymphocytes openly in a battleground, clearly those places will be the lymph nodes. Tumor cells जब अपने घर में था, not enough lymphocytes are able to see the tumor. When tumor started moving or changing their place from the breast, the first stop where they actually see lot of lymph cells will be the lymph nodes, and that in the lymph node where a lot of immune cells will be able to recognize the tumor cells, and they start showing an activity. Lymph nodes become little bit, I must say, they're little bit bigger than normal, and doctors will be able to figure out of the structure of the lymph node that the tumor cells are now leaving their place. This cancer is a metastatic version of cancer. Okay, so these are all critical information about the things. One more thing you should know about the lymphocytes is that how we able to recognize the different types of lymphocytes. Even within the T cells, there are different types of T cells. So the lymphocytes, the cells present within the lymph, they are recognized based on the presence of certain receptors on their surface, which are their markers. They have a separate markers called CD, cluster of differentiation. They have different types of receptors present on their surface. For example, thymocytes, which are also called as Langerhans cells, they have CD1 type of cluster of receptors present on their surface. It is their marker. It is a marker. It is the receptor which help us to recognize their thymocytes. The most important among all of them are CD4, CD8, and CD19. You will going to see them again and again. CD4 is the combination of receptors present on the helper T cells. It's a combination of receptor present on the helper T cell. It's also the receptor for the HIV virus. It's also the receptor for the HIV virus. Can you guess that why HIV virus is quite dangerous for human life, and why it was a trouble when it was first appeared? AIDS इतना खतरनाक क्यों था? Because the HIV virus found its partner, or it can bind itself with the helper T cell, and once the helper T cells are occupied by the viruses, the immune system, adoptive immune system, will not be able to execute its function properly, because a critical member of the adoptive immune system is attacked by the virus. तो आपने वो सुना होगा एक टर्म कि HIV एड्स में वो हमारे इम्यून सिस्टम पे हमला करता है. तो वो जो इम्यून सिस्टम पे हमला करता है, that was the CD4 cells. The receptors are also for the receptors for HIV. CD8 cells, CD8 having the combination of receptors called CD8. This combination is present on the suppressor cells as well as on the cytotoxic T cells. Cytotoxic T cells, just keep that word in your mind. The last one is CD90. This is a combination, another combination of receptors present on the B cells. So we will be able to recognize the different types of thymocytes based on the presence and absence of these receptors. So लोग इसको research
Sir, I
करते
have
हैं,
a question.
they work and help with the help of these receptors. G. Nikash.
Sir, you said that the HIV virus binds to the T helper cells, right?
Yes, it can
So,
also bind.
why,
It can. Yeah, yes, yes.
okay, so why, if it binds to the T helper cells, why the cytotoxic T cells do not detect it as an anomaly?
Why
And
cytotoxic T?
why don't they kill those T cells?
नहीं cytotoxic. Oh. No. Okay. थोड़ा सा इसको मैं adjust कर दूं. When the adoptive immune system sense a virus is there, and that virus is an HIV virus, the adoptive immune system will trigger its cascade. Adoptive immune system अपना cascade trigger करेगा. In between that cascade, there will be a time when they will going to produce the helper T cells with the CD4 type of receptors. एक वक्त आएगा जब वो क्या करेंगे, when they will going to produce an helper T cell with the CD4 combination of receptors. Those cells are vulnerable to HIV viruses. Those cells can be the target of the HIV viruses. So what that means, the adoptive immune system in general is feeling difficulties in the execution of its function, क्योंकि उनका एक मेंबर काम नहीं कर रहा. अब आप गैस करें, अगर helper T cell is not able to produce efficient, these lymphocytes, they came into existence and they get mature. So whether they are beta cells or whether they are T cells, both of them have a common precursor, and that precursor present within the bone marrow. More accurately, I can tell you that precursor cell is actually came from hematopoietic stem cells. जो हमने शुरू में देखा था, it came from hematopoietic stem cells, and after that it changes its tracks, its path. One of them enters into the stays in the bone marrow, mature over there, and convert into an immature beta lymphocytes. The other type of category, it took a different path. They convert into lymphocyte lineage, enters into the thymus, from where it can differentiate and get itself mature further to become a naive T cells or naive T lymphocytes. Both of these things are present in the circulatory system, whether they are B cells or whether they are T cells. But of course, they are designed in different way, they are created in a different way, and they are executing different jobs. Both of these cells often meet with each other, and they are connected with each other as well. They support each other well as well. For example, the infection from the tissues and from the skin and from the other part of the body, that infection was usually hunted or captured within the lymph node. This is a lymph node. Lymph node is a place where the lymph cells spend some time in the vicinity of each other. So the beta cells, when passed through the spleen, when it passed through the lymph node, it came to know whether there is any infection or not. How are they able to know that there is an infection or not? That information will bring is the job of the macrophages. Bringing that information is the job of macrophages, which are circulating, which are present in different parts of our body. So if the lymph node does not carry any macrophage with any antigen, the beta cells will feel that there is no problem, there is no need to get activated, there is no need to trigger the immune system. The naive beta cells will stay as a naive beta cells, and the process continues. Similarly, the T cells from the blood circulatory system, when enters into the spleen or lymph node or in other parts of the body, it came across nothing, it came across no antigens, it came across no macrophage with the bad thing. They will feel nothing is going on, there is no need to trigger the activity, and they will circulate as a naive T cells. The naive T cells will become a mature or active or activated version of T cells when the naive T cells is able to see or capture or catch a macrophage with the information about the bad thing. When it's able to see a macrophage with the information of the antigen. And the high chances of this meeting is possible in the structures like lymph nodes and in the spleen, where they can spend a lot of time with each other. Okay, and then otherwise they will go back to the normal things. The first thing, the first member which we are studying about the T cells, or they are also called as T4 lymphocytes or T4 helper cells or CD4 cells. ये सब नाम एक ही टाइप के cells के. It came into existence or it arises from the naive T cells. when the naive T cells came to know about the trouble, it differentiate to produce different types of T cells. One of the T cell is called as helper T cell. One of the T cell is called as T4 helper T cells. Sometimes it is called as T4 lymphocytes, and sometimes just CD4 cells. So CD4 cells का मतलब भी क्या होगा? These cells are the T4 lymphocytes or the helper T cells. They are the most specialist cells in the immune system because they are the ones who needs to read and sense not only the trouble but also needs to sense the nature of the trouble. ना सिर्फ इनको ये देखना है कि ट्रबल कौन सी है, बल्कि ये भी स्टडी करना है कि उस ट्रबल की नेचर कैसी है. Whether it is a bacteria, if it is a bacteria, then it is what type of bacteria. It's a one type of bacteria or another type of bacteria or a third type of bacteria. It should be able to distinguish between different types of pathogens as well. They are also designed to produce lot of chemicals, small proteins, which is called as cytokines. The T helper cells, they can produce a lot of different chemicals called cytokines, which can help in the stimulation and activation of the other members of the immune system. We will see कि वो chemicals कौन से होते हैं and how they help. Every T cell is carrying a receptor. Every T cell is carrying a receptor. So this is the T cell, a T4 lymphocyte, a naive T4 lymphocytes, remember. Every T cell is carrying a receptor called TCR or T cell receptor. This is not all, this is not everything. It is also supported by some other receptor called CD4. That is why it is called CD4 plus cells. The cells with CD4, the cells with the cluster of differentiation number 4, CD4 cells. So the presence of TCR is the characteristics of the T cell. The presence of TCR plus the CD4 is the characteristics of helper T cell or T4 lymphocytes. So this is something which is present on the surface of the T cell. The TCR or the T cell receptor is the receptor which is able to communicate with the MHC. There are different types of MHC, 1, 2, and 3. We will study after that, but just consider them as an MSC. The MSC which is present on antigen presenting cells, dendritic cells, last lecture. So the antigen presenting cells are carrying the MSC receptor, major histocompatibility complex, and this major histocompatibility complex is able to bind with the TCR. So the TCR or the T cell receptor is the one which can connect itself with the MHC. That is the place where communication occurs. That is the place where they can exchange information. If the CD4 is not there, that connection is not established properly. So the TCR will not able to bind with the MSC in an efficient way, and that information, the red one, will not be communicated. Think about a situation if the macrophage does not carry antigen. If this macrophage do not carry antigen, any bad thing, what will happen? This red stuff is not here. ये red stuff यहाँ पे नहीं होएगा. This red stuff means the antigen. The macrophage bringing an antigen. If the macrophage is not bringing anything, this red stuff is not there. So what will happen? When this connection is established, the TCR will going to touch an empty MSC. And that empty MSC means there is nothing in the dendritic cell. There is no information, there is no infection, there is no trouble. So the TCR will not be stimulated and the T cells will stay as a naive T cells. It will stay as naive and go away. But if the dendritic cell just met with a bad thing, it just found a bad stuff, an antigen, it is presenting that antigen on the surface with the help of MHC, what will happen? At that time, if the TCR MSC complex is established, together with the part of the antigen, together with that red stuff, now the TCR will get activated. And this naive T4 lymphocyte is no longer a naive one. It will going to be converted into a mature T4 lymphocyte. It will be different. It will be differentiated and more active and ready to do its job. Okay, so this is the role of T4 lymphocytes. But apart from this communication, it's also participate in other immune functions as well. It can share information from the dendritic cells, जैसे मैंने दिखाया. It can share information from the dendritic cell. It can also share information from the other members of the immune system as well. In the same way, it can connect with the other members of the immune system in the same way. Also, in addition to that, the T4 lymphocytes is able to generate chemicals or small proteins or cytokines, more accurately, which can activate the macrophages as well as the natural killer cells in that region. It can also produce cytokines which enable the beta lymphocytes, जो beta cells को activate करेंगी, and they will produce the plasma cells and antibodies. And also they produce cytokines which can convert the T4 lymphocytes into the next mature version, into the new cells, into the effector cells. Remember, when a T cells get mature and when the T cells get differentiated, it produces more advanced cells, and those ones are called as, generally they are called as effector cells. They are functional molecules, they are functional cells, they can execute the job. So these are the things we can relate with the T4 cells. There are different types of effector T4 lymphocytes. Effector T4 lymphocytes means, please understand these terms very accurately. The T4 lymphocytes, when get the information about the bad thing, it needs to differentiate to become a differentiated version of a cell or an effector cell. There are generally two types of effector cells based on the cytokines. One is called as Th1 lymphocytes. The other are called as Th2 lymphocytes. Both of them are T4 cells. Okay, remember, both of them are CD4 plus cells. Both of them are classified under the T4 lymphocyte. The difference between the Th1 lymphocytes and the Th2 lymphocytes is that Th1 lymphocytes, they are able to recognize antigens presented by the macrophages. They are designed to talk with the macrophages. While the Th2 lymphocytes, as I told you earlier, they can communicate with the others. Now, this is the answer, the other. The Th2 lymphocytes, they are able to talk with the B lymphocytes. Now just refresh and help your brain for this thing, that the beta cells, they are not only able to recognize the antibodies, the antigens, they have the antibodies, but they can also eat the bad thing. Beta cells भी किसी bad चीज को खा सकते हैं और उसको present कर सकते हैं. Beta cells are also antigen presenting cells. But beta cells will only able to talk with the Th2 category. It only able to talk with the Th2 lymphocyte. So full stop. If a T cell is communicating with macrophage, if a T cell is communicating with the macrophage, you can guess this is the T4 cell subcategorized as Th1. If a T4 lymphocyte is talking and communicating with the beta cell, you can guess this one is the Th2 sort of T4 cells. The other things are simple. They are different in producing different type of cytokines. For example, the Th1 cells, they are supposed to produce interleukin 2. It's a cytokine. They are supposed to produce interferon gamma. It's a cytokine. They are supposed to produce lymphotoxins as well as tumor necrosis factor beta, TNF beta. The Th2 lymphocytes, they are designed to talk with the beta lymphocytes. They produce cytokines such as interleukin 2, 4, 5, 10, and 13. And they promote the antibody production as well. So these are the little small molecules which help them to understand what type of job they need to execute. Remember, if a disease is in a situation, where a lot of antibodies are required to be produced, अगर बहुत सारी antibodies चाहिए होंगी, so you can guess that out of the T4 cells, the Th2 types of cells are working more as compared to the Th1 types of lymphocytes. Which one of them is acting more than other depend on the nature of the trouble and the function of the immune system at that time. The other cell is the T8 lymphocytes, the CD8 cells, or I alternatively called it as cytotoxic T cells, if you remember. They display the CD8 molecules on their surface. They display the CD8, the cluster of differentiation combination number 8, T8 lymphocytes. They are different than the T4. They are a little different than the T4, the typical T4. The T8 cells, please stay with me, okay? I'm sorry, it's going to be lot of information in a small period of time. Sorry, okay? Just thoda sa, but this is the last thing I want you to understand. The T8 lymphocyte is different from the T4 because the T8 lymphocyte specifically able to capture or talk or communicate with MSC1 type of receptor. MSC1, ये बहुत अहम और जरूरी बात है. If you remember the earlier slide, the TCR is communicating with MSC2 generally, that is a T4. But if in case of T8 lymphocytes or CD8 cells, they can communicate with the MSC1. हम पढ़ेंगे आगे कि MSC1 और MSC2 में क्या फर्क होता है, but I can tell you in a easier terms at this time that MSC1 is there in case of viral infection. MSC1 की जॉब होती है तब जब वायरल इन्फेक्शन होता है. तो आप गैस कर सकते हैं कि इन केस ऑफ अ वायरल इन्फेक्शन द MSC1 इज द मेजर player which is communicating information to the CD8 cells. Cytotoxic cells might be the major player. So T8 lymphocytes, they're genetically programmed. They produce the TCR, but that TCR is special. That TCR can bind with the MSC1, not with the MSC2. They are designed to capture or communicate through MSC1 complex. It's a body's major defense against the viruses. You should be able to guess this thing now. जो मैंने पहले कहा था कि ये हमारी body का major defense against viruses and intracellular bacteria, and particularly the cancer cells. They are the ones they can kill the tumor cells. They are the ones they can kill the bad cells which are not behaving in a normal way. Very important ones. If we are able to control them, if we are able to control the function of the CD8 cells, we can remove and eradicate the dysfunctions. And I can tell you in the near future, अगर cancer का कोई इलाज आएगा, it came from these CD8 cells because they are the ones which can kill the other cells, other bad cells as well. And then the last one are the beta cells. I want to show you a video just to get yourself relaxed a bit, because I don't want you to have that much things in your head. Any question? अभी लेक्चर खत्म नहीं हुआ, ओके? अभी और हिम्मत और बहादुरी के साथ रहना है. Any question?
सर मुझे एक कंफ्यूजन है कि अब मुझे पता नहीं है मैं ठीक कह रही हूँ या नहीं, आप मुझे करेक्ट कर सकते हैं, बट मुझे ये समझ
कोई बात नहीं.
ठीक है, मुझे ये समझ आई थी जब बॉडी में फॉरेन कोई बॉडी एंटर करती है तो डेंड्राइटिक सेल्स टी सेल से कम्युनिकेट करते हैं और टी सेल पे डिफरेंशिएट हो जाते हैं साइटोटॉक्सिक हेल्पर सप्रेसर और मेमोरी सेल्स में.
ठीक है.
तो सर ये फिर दोबारा से हेल्पर टी सेल्स भी आगे से दो डिफरेंट टाइप्स के हैं, वो दोबारा कम्युनिकेट करेंगे, क्यों हो रहा है फिर दोबारा?
फर्स्ट थिंग फर्स्ट, जो आपने पहले सुना है समझा वो ठीक है. वो ये है कि जब कभी एक ट्रबल आएगी बॉडी में, व्हाट विल हैपन, दैट विल
डेंड्राइटिक
सेल्स कैप्चर करेंगे एंड देन कम्युनिकेट करेंगे other members of the immune system, particularly the T cells. Okay? Naive T cells. The naive T cells becomes different types of T cells. One of them is T4.
Okay.
The other one is the CD8.
Mm-hmm.
Okay, now tell me it will going to be the T4 or CD8. कौन होगा दोनों में से कि दोनों होंगे? It depends on the nature of the pathogen.
Oh.
Let's suppose if the trouble is
Oh.
like viral. For example, अगर ये virus की trouble है, it is captured by the dendritic cells which are producing MHC1. अभी हमने पढ़ना आगे जाके. MHC1 तो T4 से communicate ही नहीं कर सकता. MHC1 किससे communicate कर सकता है? CD8 के साथ. अभी पढ़ा हमने थोड़ी देर पहले, है ना?
जी जी जी.
So that will going to trigger the cytotoxic T cells.
अच्छा.
So why different types of responses are possible from the immune system? It depends on the nature of the pathogen, and that is how the difference is controlled through the receptors. देखो MHC receptor रखने वाले cells virus को पकड़ सकते हैं. Okay? ये I am just telling you in a easier word. हम आगे डिटेल से पढ़ने वाले हैं. MHC1 carrying things can capture the virus. So that means the MHC1 stuff has the information about the virus, has the information about the tumor cells. So MHC1 ये information किसको दे सकता है? It can only pass on that information to the CD8 cells.
Beta cells.
CD8 cells.
Oh yes yes.
अभी पढ़ा हमने.
CD8 T cells तो नहीं ना memory cells बनते.
नहीं memory cells बनते हैं, T cells भी बनते हैं. Is one of the.
वो तो बीटा सेल्स नहीं डिवाइड करते प्लाज्मा सेल्स
भी मेमोरी सेल्स बनते हैं. टी सेल्स के भी मेमोरी सेल्स होते हैं, बीटा सेल्स के भी मेमोरी सेल्स होते हैं. ठीक है?
ऑलराइट.
शाबाश. तो अब एक काम करते हैं. ओके, लेट मी शेयर यू अ स्मॉल वीडियो एंड आई टोल्ड यू रिमेंबर द नेचर ऑफ द सेल्स. आपने आंख रखनी है इसके ऊपर कि ये जो है किस किस्म का जो है वो इन्फेक्शन है, किस किस्म का ट्रबल है. ओके, लेट्स स्टार्ट. Okay, guys. Anybody wants to summarize this video? Free scores.
So there were a lot of different receptors जिनके बारे में अभी तक पढ़ा नहीं है.
क्या जेन में आया है? मैंने इसलिए कहा था, हम ये इसको हम पढ़ेंगे ना आगे की चीज. मैं अब जान के इंट्रोड्यूस कर रहा हूँ आप लोगों को, जरा नजर आ जाए रिसेप्टर्स के बारे में. बट देयर इज नो नीड टू रिमेंबर द रिसेप्टर्स.
Sir, I
एनीबॉडी
guess
वांट्स टू
the
टॉक? मैं चाहता हूँ
mechanism,
आपको क्या लगा, बस आप अपनी समझ शेयर करें मेरे साथ.
sir, I guess the mechanism is different for a benign tumor and a cancerous tumors. The benign tumors are easy to break down and cancerous are not.
Okay, anybody else?
Sir, if we block CTLA4, then and we let the
Shabash.
T cells work against the tumor, then
Okay,
there is
good.
also a possibility that there may be a heightened reaction and there no attenuation may take place. So could it lead to adverse effects?
Okay, good. Okay, just hold this video with you, okay, for some period of time. We will talk in the next lecture about it, okay? Here we go.
Okay.
If you can see my screen once again, I will going to talk about the beta cells. So the beta cells, beta lymphocytes, they are produced in bone marrow. They mature in the bone marrow. Their job is to do a lot of things. The primary job which we remember about the beta cells is that they convert into plasma cells, they produce plasma cells, and plasma cells produce antibodies. That's something which we know. So when beta cells are produced, they are developed, they are designed to do several jobs, including that they can release certain cytokines. They can communicate, they can talk with the other cells as well. They are genetically programmed, like with the help of gene splicing, to produce different types of antibodies. They can like create so many different variety of antibodies. And the antibodies present on the surface of the beta lymphocytes, whatever the things present on the surface of the beta cells, they serve as a receptor. And these are called as surface immunoglobulins, SIGs. So whatever the proteins, whatever the receptors present on the beta cells, they are surface immunoglobulins. How beta cells get activated? They can get activated by two means. The one, without the support of T cells, T independent antigen, without any support of T cells. And they can activate with the help of the T cells. Most of the time, they get activated with the support and help of the T cells, because the T cells are carrying specifically information about the bad thing. The T cells receive that information from the dendritic cells, and then they become differentiated to produce effector cells. One of the effector cell is called as helper T cell. That helper T cells can talk, can communicate with the beta cells. How? They can release cytokines, which can be absorbed, received by the beta cells. And if the beta cells interact with the T4 lymphocytes, that beta cells will going to activate exponentially. I can tell you, T-dependent activation of beta cell is very fast, is very efficient. अगर beta cell को T cell मिल गया है, which is not a naive T cell, so then this beta cell will going to activate at a very faster rate. But if there is no T4 lymphocytes, if there is no helper T cell over there, the beta cells still can execute their function. It's called T-independent antigen. Some of the carbohydrate, lipids present in bacteria and other things, they can be captured by the lymphocytes, and lymphocytes can put them, and lymphocytes can activate an antibody response against those small little molecules without the support of T cells. But that antibody response is limited. That is a very basal level of antibody response. The beta cells will use all of its energy to produce antibodies when when it is supported and communicated by a helper T cells or T4 T cells. If you can compare these two things together, अगर आप इन दोनों को एक ही वक्त में इमेजिन करके देखें, ये बहुत I think interesting information हो जाएगी आपके लिए. Why the beta cells is acting exponentially when it is touched by the beauty of T4 cells, and why, if the trouble is there like that, but there is no helper T cell, there is no T4, there is no CD4 cells over there to support and to activate the beta cells, the beta cells will still do their job, but not with the same potential. The reason, anybody wants to tell me about that thing, the reason is that, I can help you. The reason is that the presence of CD4 cells, the presence of T4 lymphocytes indicates that there is a big trouble going on within the body. Because the T4 cells, the creation of T4 cells is resulted out of a cascade of events which has been started somewhere else. कहीं पर कोई बैक्टीरिया था, कोई ट्रबल थी जिसको मैक्रोफेज ने खाया, उसने फिर कम्युनिकेट किया टी सेल्स को, नाइव टी सेल्स मैच्योर हुए, डिफरेंशिएट किया, प्रोलिफरेट किया, प्रोड्यूस the CD4 cells. The presence of CD4 cells let the beta cells know that there is a serious activity going on somewhere, and you need to respond accordingly. So that is why a stronger response is required from the beta cell at that time. T dependent activation of beta cells. On the other hand, if there is no big infection over there, there are no CD4 cells over there, so the beta cells can find something bad. Beta cells को कुछ बुरा मिला है. Beta cells can consider that thing as a bad thing. Beta cells can produce antibodies against that bad thing, but it is not considered as very serious threat because of the absence of CD4 cells. CD4 cells की मौजूदगी बताती है कि something is like something huge is going on over there. Okay, so let me conclude this thing for today. If you able to decipher this slide comfortably, that I will be happy कि हमने कुछ थोड़ा सा एक step बढ़ के सीख लिया. ये it's like a summary of everything in a nutshell. So this is the helper T cell. This one is the helper T cell, CD4 helper T cells. You can guess it's at the receptor. इस receptor का नाम क्या होगा? This receptor name is called TCR or T cell receptor. This receptor can communicate with the receptors present on the beta cell, and there are some supporting receptors as well. Once this connection is established, the T cells produce cytokines. याद रखें कभी कभी cytokines को lymphokines भी कहते हैं, घबराना नहीं चीजों से. Lymphokines general नाम है ना, cytokine भी general नाम है. Some chemicals, small molecules, peptides, which can activate the beta cells. Once this connection is established, helper T cells releases cytokines or lymphokines which activate the beta cells. Activation of beta cells results in the production of more antibodies as well as convert them to produce plasma cells. The plasma cells are designed to secrete huge response of antibodies in the systems. So that is called as the T dependent activation of beta cells. If this thing is not there, if the helper T cell is not there, beta cells is still able to produce these antibodies and to capture some bad things present in the cells. But that is not a primary response. That is not something very big. A big action is required when something actively going on in the body, and that can only be sensed with the help of these helper T cells. Please leave your name and ID, okay, in the chat box. In the meanwhile, I conclude that thing. Okay. Let me leave the name and ID. Okay. And then the last one, the natural killer cells. They are the members of the innate immune system. They are very special. They can kill the cells. Oh my God, they are the lymphocytes. They can kill. They lack beta cell receptors and T cell receptors. So that means they cannot talk to them. They are designed to kill certain mutant cells or virus infected cells. They usually communicate with the help of certain receptors, and they are able to kill the cells. Especially, they kill the cells who don't have the markers of identity, marks of identity. Before it finished, it's about to finish, so I will talk about these things, how nature killer cells distinguish between our own cells from the bad cells. Okay, so my lecture, here we go. I can tell you that we have built our foundation about our concepts related to molecular immunology. That how many players are there? That's the first thing we should know. The second thing we should know is that which player comes at what time during the battle. This is quite critical and important. And the higher level of understanding is that which of these players are dependent on each other, in what way, who is dependent on who, something like that. If you have these ideas, you should be able to decipher most of the textbooks related to molecular immunology. This is my personal suggestion, but since I'm a professor, so this is my advice to you guys, that if you are learning from me, I might not be the best way to test whether you are getting the things in a perfect way or not. I might not be the best way. The best person or the best way is like to test your knowledge and skills at other places. So whether I will going to give you any task or not, whether I will ask you to do these things or not, please try to test the standard of your understanding about molecular immunology in browsing and reading some pages from some other textbooks of molecular immunology. Just try to spend some time like reading a couple of pages to check whether you are getting the things out of that page. When you will be reading the research papers, remember it's a little bit different world, because the research paper also includes lots of techniques and many methods which you haven't heard during your lifetime. So that is absolutely fine not to get everything out of the research paper when you read that paper and material for the very first time. ये बिल्कुल होएगा, ये हम सब के साथ होता है, मेरे साथ भी होता है. That is nice, but you should be able to feel when you are reading. जैसे होता है ना कि यार ये जगह देखी है, ये पहले कहीं हुआ है मेरे साथ. I know all these things. So please, I request you to test your skills, to test your concepts about molecular immunology, because if you are like studying a research paper which explains that how T cell recognize and process the information about the antigen, so you have to go through the molecular details about that study. You have to go through the methods the authors are using, they have used to decipher that mechanism. But you already know the mechanism, you already know the answer, that T cell process that information. This is like an understanding of an undergrad student. Now what will going to make you a different person is that you should know that how researchers are able to decipher these signaling cascades, how they able to decipher, what are the methods they are using in order to in order to unveil these mechanisms. वो research papers में होती हैं सारा material. I request you to please stay very close to those things. वो बहुत मुश्किल और uneasy और कहते हैं ना अबाजार किस्म का काम है, but that will going to make a difference between you and the Allah ना करे, between the people who are successful or who are less successful. इन दोनों में जो फर्क करेगा in terms of रिसर्च वो यही है, जो अच्छा पढ़ते हैं साइंटिफिक मटेरियल्स को, और वो लोग जो नहीं पढ़ते हैं। रिमेंबर, आपका नॉलेज यूट्यूब के ऊपर डॉक्यूमेंट्रीज देखने की वजह से, मटेरियल्स और न्यूज़पेपर रीड करने की वजह से, लिटरेचर रीड करने की वजह से, ये आपके साइंटिफिक स्किल्स को नहीं बढ़ाता है। ये बहुत बड़ी गलत फहमी है कि यू नो अबाउट द स्टार्स, you know about the astronomy. That is nice, but you are not a scientist, so you don't have the technical information how to measure these things. So as a researcher, as a scientist, you should be different from the other person. Most of the people know that the T cell will going to decipher the nature of antigen, and then it will going to process one antigen in a different way and another antigen in a different way. Everybody knows. It is easy to understand. But as a researcher, you should try to dig that how a oligosaccharide is sensed differently by the T cell, differently as compared to the protein. जब T cell किसी प्रोटीन को कैप्चर करता है, सेंस करता है, तो वो उसको अलग तरीके से प्रोसेस करता है। जब T cell किसी ओलिगोसैकेराइड को फील करता है, तो वो उसको अलग तरीके से प्रोसेस करता है। How it is possible at the molecular level, and what are the things involved? And if you need to process, if you need to execute those experiments, what are the skills you need to have? कौन-कौन सी ताकत और सलाहियत जो है वो आपको इख्तियार करनी चाहिए? Please work on these things to make yourself a stronger and a better candidate. Okay, I will going to share my screen with you, and I will going to talk about the last couple of slides of this lecture. After that, we have finished our lecture number 3 and 4. Traditionally, this is like a half of our syllabus. This is like the first half of your molecular immunology course, and after that we will going to spend like few days or couple of days on one topic. After that, this is like the conclusion or the end of our first part of molecular immunology, where we have completed the picture. We have studied about the beta cells, and that is a simplified version that how these cells, especially the beta cells, are executing their job in terms of infection. Remember, the beta cells are quite amazing in their function. They can capture the antigen. वो bacteria को भी खा सकते हैं. They can capture the antigen. They can present the antigen. उसको present भी कर सकते हैं T cells. And also, the beta cells, they are also carrying the antigens. उनके पास antigens भी हैं. एक तो ये antigen presenting cells का काम कर रहे हैं. At the same time, they are also carrying the different types of, you must say, receptors. Beta cell receptors or PCR. Did you remember T cell receptor, the TCR? It's quite general term. Like any antigen presenting cells, beta cells can communicate with the helper T cell. If you remember, यहाँ पर macrophage को होना चाहिए. If you remember, इस जगह के ऊपर macrophage को होना चाहिए, इस जगह के ऊपर, because macrophage uses major histocompatibility complex to communicate that information with the helper T cell. Actually, with the naive T cell, sorry, not with the naive T cell. अगर आपने ये बात हमने पहले पढ़ी हुई है, helper T cells कि नहीं. Let me repeat to remove confusion. Earlier you have studied macrophages can interact and present the antibodies to the naive T cells. To the naive T cells. Macrophages जो हैं वो naive T cells को information pass on करते हैं. It's a similar sort of thing. Beta cells are communicating information to the helper T cell. And remember, helper T cells की मौजूदगी किस बात की तरफ इशारा है? अगर आप इन दो प्रोसेस को कंपेयर करें, मैक्रोफेजेस नाइव टी सेल्स को बता रहे हैं, ये इनिशियल शुरू के दिनों में होगा, इनिशियल डेज में होएगा, जब कभी मैक्रोफेजेस बताएंगे नाइव टी सेल्स को पहली मर्तबा कि देखो इस दरिया के दूसरी तरफ भी एक दुनिया है, देखो दुनिया जो है वो इससे आगे भी कहीं, इससे आगे भी है, लेट्स मूव ऑन, देयर इज समथिंग weird going on in our body. So for the very first time, when macrophages came to know about the trouble, they communicate with naive T cells. It's like it might be doing in the second day or the third day of infection. जब शुरू होगा तब ये काम हो रहा होगा. Naive T cells उसके बारे में जानेंगे, सोचेंगे, they get educated, and after that they proliferate and produce different effector cells. One of the effector cell is called as helper T cell. उनमें से एक इफेक्टर टी सेल कौन सा होगा जिसका नाम है हेल्पर टी सेल. और हेल्पर टी सेल वो होता है जो बीटा सेल्स को सपोर्ट करता है, बीटा सेल्स को पुश करता है, उसको साइटोकिन्स देता है, उसको प्रोलिफरेशन करने में, उसको एंटीबॉडीज प्रोडक्शन में सपोर्ट करता है. दिस इज द हेल्पर टी सेल वी आर टॉकिंग अबाउट. इफ यू आस्क मी सर ये वाला हेल्पर टी सेल कहां से आया, तो ये वाला हेल्पर टी सेल दिस केम फ्रॉम द सेम मैकेनिज्म आई हैव शेयर्ड. ये कहां से आया होगा? दिस हेल्पर टी सेल केम फ्रॉम द नाइव टी सेल. नाइव टी सेल को पता कैसे लगा? फ्रॉम अ मैक्रोफेज, फ्रॉम समवेयर एल्स. द नाइव टी सेल्स converted, differentiated and produce helper T cells. Now this helper T cell is communicating with the beta cell on this slide. This helper T cell is communicating with the beta cell. Now you can tell me, if the helper T cell is communicating with the beta cell, it is pushing the beta cells to go further and to produce the plasma cells and to produce the antibodies to kill the trouble. But conversation between a beta cell and a helper T cell means that the trouble was already there in the system. क्योंकि helper T cell नहीं बन सकता, helper T cell नहीं बन सकता जब तक naive T cells इस problem के साथ मिल ना चुका हो. So the communication between the beta cells and helper T cell means that the issue is greater, is bigger, because the helper T cells are there, infection was there. Infection होएगा तो helper T cell बना. So since the problem is big, so the helper T cells are there, so helper T cell will push the beta cells to produce more and more antibodies, and that will going to kill the trouble. So that is how the different levels of trouble can be assessed, can be estimated by our immune system. First करते हैं, first phase में, if the problem is not big enough, the naive T cells came to know about the problem from the macrophages, but the problem is so small that the macrophages has consumed it. Macrophages ने खत्म कर दिया, kill कर दिया problem को, और adoptive immune system trigger करने की जरूरत नहीं है. थोड़े बहुत helper T cell बनते हैं, लेकिन major helper T cell नहीं बनते. If the helper T cells are not there, naive T cells बहुत सारे helper T cell नहीं बना पाएगा, या helper T cell बना ही नहीं पाएगा. What will happen? In the absence of helper T cells, beta cells will not going to be proliferated, beta cells will not going to produce plasma cells, beta cells will not able to produce enough antibodies, क्योंकि उसकी जरूरत ही नहीं है. So nobody will push the beta cells to produce antibodies. Pushing beta cells to produce antibodies is very costly, it's a very high expensive process, it will consume a lot of energy of our body. So ये तभी हम करेंगे जब हमें पता हो कि the trouble is there, it's a big trouble, the problem is really big. So this slide actually summarizes that how these two cells, beta cells and T cells, they got activated. Both of them were naive. Once beta cells were naive, and once both of them were naive, they never came across any trouble. They travel in the bloodstream. They move into the secondary lymphoid organs, like they used to stay within the lymph node. They used to stay within the spleen, and these two are the places where they came to know about the problem. इन्हें problem के बारे में कौन बताएगा? Who will let them know about the trouble? The other cells like macrophages, dendritic cells, antigen presenting cells, they will tell them about the problem. The naive T cells are only able to get the things, to get the information from the antigen presenting cells. The beta cells, on the other hand, they can directly capture the bad thing. They can directly capture the bad because they are antigen presenting cells as well. So once the red ones are the T cells. Once the T cells came to know about the problem, they will going to produce the effector cells. They will proliferate, differentiate, and produce the effector cells. There are different types of effector cells. Effector is a term generally applicable. There are different types of effector cells. One of the type of effector cell is called as helper T cell. उनमें से एक का नाम है helper T cell. And the job of the helper T cell is to push the beta cells to produce more antibodies. So effector cells will be produced together plus क्या होगा साथ में? They will going to push the beta cells to produce lot of antibodies. So here you can see effector cells are there as well as the antibodies are there. That does not mean effector cells are producing antibodies. Okay? Antibodies कौन पैदा कर रहा है? Beta cells पैदा कर रहे हैं. Antibodies ये blue ones पैदा कर रहे हैं. But the beta cells will produce lot of antibodies कब? When it is supported and pushed by one of the effector cell, which is called as helper T cell. जब helper T cells beta cells को push करेंगे, तब ये काम. For example, let's start from here. For example, this is the journey of the trouble. If an antigen or a part of the bacteria manage to enter into the skin, it is captured by a cell from the from antigen presentation from the macrophage or dendritic cells from the skin. It is captured, and then it is taken away into a secondary lymphoid organ. That is the place where body came to know about this thing. That is the place, the lymphoid, secondary lymphoid organ, where the dendritic cells are able to present or to share these things with the T cells and the beta cells. There is another chance. There is another way. Another way is that the antigens and microbes, they can manage to reach to the blood. If they are present in the skin, it will be captured by the macrophages present in the skin and taken to the lymphoid organ or lymph nodes. If the bacteria or the microbe or the virus is managed to enter into the bloodstream, it will not be captured by the macrophages. It will go to the spleen, and in the spleen it is captured. In the spleen we came to know about this thing, and then we produce effector cells and the antibodies, and these effector cells and antibodies will going to show their response and will go back into the blood, will stay in the blood, and will start killing the microbe, bacteria or virus present within the blood. So from the skin, it is taken by the dendritic cells, presented to the immune cells within the lymphatic system or within the lymph node. From the blood, it is sensed within the spleen. So if the person do not have a spleen in certain cases, if the spleen is not working well, that person might not be able to detect the germs and problems present in the blood. But that person might be fine with the problems present in the skin, but not be fine with the problems present in the bloodstream. Sometimes, in some cases, the spleen, which is responsible to detect the pathogens present in the blood, miscalculating about the problems. Sometimes the spleen miscalculating about the trouble. It started responding to the things which they should not. It started considering something bad. It started considering like something which is not supposed to be bad as a bad. If the spleen started making wrong calculations about the things present in the blood, क्या हो अगर वो अपने white cells के बारे में बुरा सोचना शुरू कर दे? क्या हो अगर वो अपने platelets के बारे में बुरा सोचना शुरू कर दे? If the spleen started making wrong calculations about the things present in the blood, what will happen? It will push our immune systems to start working against our own blood, start working against the things present in our own blood. So if the spleen is overreacting, keep pushing our immune systems, that is a very big trouble as well. And in those cases, the spleen is required to be removed. वो कहते हैं कि spleen को remove करना पड़ेगा in those cases, so that the body's immune system should not be overreacting. The last thing you should know, that is about the natural killer cells. They are present both in innate as well as in adoptive immune system. जी 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 शाबाश. Any question?
Sir, can I ask a question?
Why not? Why not? Why not?
सर ऐसा होता क्यों है, लाइक व्हाई डज दिस हैपन कि स्प्लीन अपने ही मतलब इम्यून सिस्टम के खिलाफ मतलब वो काम करना क्यों स्टार्ट कर देता है? व्हाई डज इट कंसीडर इट्स ओन ब्लड एज फॉरेन बॉडी? लाइक व्हाई डज दिस हैपन? इज देयर अ स्पेसिफिक रीजन फॉर दिस? एंड आफ्टर द रिमूवल ऑफ स्प्लीन इज द पर्सन फाइन? लाइक for example, if someone has got their spleen removed and in the current situation of COVID, so is there probability of capturing and getting COVID more than
Yes.
the normal person?
So two questions. The first answer is that there are many reasons which can lead to this situation. The first thing is that there might be genetical reasons, that the combinations through which a spleen is detecting the problem are not working nice, were not engineered correctly. So we don't have that combinations right. तो वो गड़बड़ करते हैं. Genetical reasons हो सकती हैं. Of course, remember, the way we detect the problem is a way of calculating things based on the probability. Probability is a probability. Although if you are 99.9% sure in making the right calculation, there is always a chance, even if it is less than 0.1%, to make wrong calculations. एक तो ये वजह होती है. But there are other things, कि sometimes the microbe, the bacteria, it is structurally so similar to your own cells, or structurally so similar to your own cells that your spleen or your immune system fail to distinguish between the good things and the bad things. ये वजह हो सकती है. लेकिन most of the time जब spleen काम नहीं करता, वो genetical reason से होता है. There are other factors as well. It's not all, but these are the factors which push the spleen not to work in a way it should. If you remove the spleen,
I have a question as well.
मैं second part का answer कर दूं. If you remove the spleen, what will happen in that case? If you remove the spleen, you are actually taking a risk, a calculated risk, that overactivation of spleen is causing more damage to your body. Because if somebody push your immune system to be used every day without any reason, your resources will be exhausted. Person will be dead. I mean, it's very difficult for that person to survive with an over reaction, with an over responsive immune system. तो डॉक्टर ये सोचते हैं कि ये जबरदस्त बंदा ठीक है, लेकिन इसका spleen started fighting against the things present in his own blood. So it is better to remove the spleen ताकि over reaction को रोका जा सके. So they make a very difficult calculation about it, pushing the limits, that expecting that the removal of spleen will allow your body to have a lesser evil. अगर आप मुझसे पूछें कि उसके ब्लड स्ट्रीम में और जर्म्स आएंगे, तो आंसर इज यस, दे डोंट हैव चॉइस. तो उस आदमी के पास ये चॉइस है कि वो या तो बीमारी से दूर रहे, और अगर बीमारी उसके पास आए तो मैनुअली किल करे. मैनुअली किल करने का मतलब समझते हैं ना, डॉक्टर की मदद से मारे उसको, इंजेक्शन और दवाइयों की मदद से मारे. जी.
Sir, is there a possibility that all of these autoimmune diseases are caused by this misinterpretation and miscalculation of spleen?
Yes. Miscalculation has a big meaning. It has a big meaning. देखो ये भी तो miscalculation है ना. You capture something which is not toxic. If the macrophages started capturing something which is not toxic, miscalculation है ना. ये भी तो miscalculation है कि if your TCR will sense something which is less bad, your TCR will start sensing it as a very very bad. Miscalculation, overreaction है.
But sir, ये सिर्फ spleen की वजह से होती है?
नहीं. Spleen is one of the example. But remember the difference between the spleen and the other thing, कि spleen blood में अगर कोई microbe होगा वो बहुत बड़े समंदर में होगा. अगर खून में कोई बैड चीज मौजूद है तो वो एक ओशन में है. फ्रॉम दैट ओशन टू एक्सट्रैक्ट द राइट थिंग्स, कहां से पता लगेगा इसके अंदर जर्म्स का? तो स्प्लीन इज अ प्लेस जहां से जब वो खून गुजरता है तो इट कैन बी फिल्टर्ड एंड इट इज इंटरेक्टेड विद द अदर मेंबर्स ऑफ द इम्यून सिस्टम. तो अगर स्प्लीन में गड़बड़ होगी तो ब्लड से पिक करना मुश्किल हो जाएगा बैड सेल्स को. ठीक है?
So,
सर
sir,
मेरा
there is a
क्वेश्चन
जी शाबाश.
सर मेरा क्वेश्चन ये है कि जब बंदा सेप्टिक शॉक में चला जाता है या सेप्सिस में चला जाता है तो देन इज देयर अ प्रॉब्लम विद द स्प्लीन या स्प्लीन उसे डिटेक्ट नहीं कर पाती है क्या? व्हाई डज द डॉक्टर से कि अब ये सेप्टिक शॉक में चले गए हैं तो इट्स हार्ड फॉर हिम टू कम बैक और हार्ड फॉर हर टू कम बैक?
सेप्टिक शॉक से क्या मुराद है आपकी? आप क्या कहना चाह रहे हो? काइंडली इसको एलैबोरेट करें.
सर सेप्सिस मतलब एज एन बैक्टीरियल इन्फेक्शन बहुत ज्यादा हो गया है ब्लड में.
ये स्प्लीन की वजह से हो सकता है. यू राइट. और ये वही है जो हम डिस्कस कर रहे हैं कि अगर स्प्लीन नहीं काम करेगा तो ब्लड में मौजूद बैक्टीरियल, फंगस और एनी अदर सॉर्ट ऑफ इन्फेक्शन विल नॉट बी एबल टू फिल्टर इट. हम उसको पकड़ नहीं पाएंगे. तो डॉक्टर्स को या आपको मैनुअली खुदा ना खासा इसको डील करना पड़ेगा. इट्स नॉट अ सॉल्यूशन, इट्स अ लेसर इवल. ठीक है? डू वी हैव मोर क्वेश्चन?
yes, sir.
जी
Sir, so
शाबाश.
there is a disease called immune thrombocytic purpura. It's a blood disease where your platelets start getting very low, like it's less than even thousands at times. So doctors suggest that it's either going to be fine in 6 months or otherwise it is going to be chronic and you have to get your spleen removed. So my question is that is there no other way for autoimmune diseases, especially related to blood, and there is no other solution other than removal of spleen?
हमने एक बहुत अहम क्वेश्चन को भी एड्रेस नहीं किया. बहुत अहम क्वेश्चन को वो बड़े एंड में जाके करेंगे. वो बड़ा खूबसूरत सवाल है. यानी कि हमारा ये जो ऑटोइम्यून डिजीजेस और इससे रिलेटेड क्वेश्चंस है ना, ये सारे उससे आ रहे हैं. अच्छा, ये देख लेना, ये लास्ट स्लाइड है. इट इज लाइक दे आर डिजाइन्ड टू किल द सर्टेन म्यूटेंट सेल्स एंड इन्फेक्टेड सेल्स. नेचुरल किलर सेल्स जब उसको मिलते हैं तो दे आर एबल टू किल द सेल्स. मैंने इसके बारे में बात की हुई है और ये मैं शेयर कर दूंगा यूट्यूब वीडियो और आप कोई और भी देख सकते हैं. सो इट्स नथिंग स्पेशल. लेट मी आंसर द क्वेश्चंस. मोस्ट ऑफ द थिंग्स व्हिच आर रिलेटेड टू द ऑटोइम्यून डिजीजेस, उनका ताल्लुक एक बहुत सेंसिटिव किस्म के मैकेनिज्म के साथ है. दैट सेंसिटिव मैकेनिज्म इज associated when our immune cells started learning about the self and the non-self for the very first time. मैंने इसमें बहुत टाइम लगाया शुरू में जानबूझ के, because I wanted you people to understand very clearly the self and the non-self, and especially जो मैंने अभी नहीं पढ़ाया, that how our immune cells came to know that this is the cell which belongs to me. Even between you, आपका स्किन मेरे स्किन में ग्राफ्ट करेंगे तो my immune system will start fighting against your skin cells. थोड़ा सा भी करेंगे तो खराब हो जाएगा, वहां से बहुत बुरा होगा. Keep fighting and killing that external skin graft. Although have the same
Sir, I
epithelial,
have a question.
although have the same epithelial cells, same genome, same structural proteins, everything is same. Still, my body, some of the immune cells will start feeling this is not mine. जी, क्या क्वेश्चन है? मैं अभी
Sir, जो
इसको हाईलाइट करता हूँ. मैंने अभी काशिफ कंक्लूड नहीं किया. मैं आपको इशारा दे देता हूँ थोड़ा सा. इसको हम खास तौर पे पढ़ेंगे. मिसेस को सवाल किसने पूछा था पहले? मुझे नाम भूल गया.
Sir, Naiba.
नाइबा. तो नाइबा, मैं इसका आपको हिंट दे देता हूँ. ऑटो इम्यून डिजीजेस का जो ओरिजिन है, जो ओरिजिनल सिन है, वो कहाँ रहता है? जब हमारी बॉडी फॉर द वेरी फर्स्ट टाइम इन फॉरएवर develop the immune cells during the development, or even in the during adult cells, we are producing new cells ना. हम naive T cells पैदा कर रहे हैं, हम naive beta cells पैदा कर रहे हैं. जब हम cells को पैदा करते हैं ना, हम इनको टेस्ट करते हैं against all the antigens which we are having. Means हम इसको इंट्रोड्यूस करते हैं against all the self. Okay, यानी कि ये ऐसे बदतमीज सेल्स होते हैं which are designed to fight against एंटीजन। हम उन तमाम नए पैदा होने वाले बदतमीज सेल्स की लाइब्रेरी को एक्सपोज करते हैं to all the antigens present in our body। हमारी बॉडी में जितनी भी एंटीजन उनसे इंट्रोड्यूस करते हैं, बताते हैं कि ये खालू है, ये फूफा है, ये चाचू है, ये भतीजा है, ये दादाबू है, ये पापा है, ये फलाने हैं, ये फलाने हैं, इन सब से इंट्रोड्यूस करते हैं वन बाय वन। इफ यू आस्क मी एट दैट टाइम आवर इम्यून सेल्स क्या रिस्पॉन्स होगा उनका अपनी एंटीजेंस के खिलाफ जब वो इंट्रोड्यूस कर रहे होंगे पहली मर्तबा, तो क्या करेंगे वो? जब वो अपने एंटीजेंस को मिल रहे होंगे, उनमें से बहुत सारे इम्यून सेल्स जिसका कॉम्बिनेशन मैच करेगा वो रिएक्ट कर जाएगा अपनी एंटीजन से। ऐसे मिलाएंगे हम, इससे मिलो, ये है दादाबू, तो एक इम्यून सेल विल स्टार्ट फीलिंग एक्टिवेटेड, will started feeling reactive against दादाबू। तो हमारी बॉडी उस वक्त ये सिलेक्ट करेगी कि इस एक इम्यून सेल ने हमारे एक अपने भाई के खिलाफ, अपने एक मेंबर के खिलाफ, अपने एक सेल्फ के खिलाफ रिस्पांस शो किया है, इट शुड बी किल्ड, उसको मार दिया जाएगा। यानी कि शुरू में जब इम्यून सेल्स की लाइब्रेरी बन रही होती है बहुत सारी, उनमें से हर वो सेल जो सेल्फ के अगेंस्ट हल्का सा भी रिएक्शन शो करता है, उसको डिलीट कर दिया जाता है, उसको मार दिया जाता है। आर यू विद मी? आप मेरे साथ हैं? जरूरी बात कर रहे हैं हम बहुत ज्यादा। ओके, नेम एंड आईडी लिख लेना जब मैं आपसे कहूंगा तो। ओके, बोलो, मेरे साथ रहना प्लीज। सो फॉर द वेरी फर्स्ट टाइम व्हेन वी आर लाइक हैविंग लॉट्स ऑफ न्यू सेल्स, दो सेल्स वर एक्सपोज्ड अगेंस्ट आवर ओन एंटीजेंस। उनमें से अगर किसी ने सेल्फ एंटीजन के खिलाफ हल्का सा भी रिएक्शन दिखाया, उसको मार डालते हैं उधर ही। Whatever the few cells we left, जो थोड़े से सेल्स बचते हैं ना हमारे पास, वो सेल्स कौन होंगे? वो सेल्स किसी चीज के खिलाफ भी रिएक्ट करेंगे, लेकिन नहीं करेंगे तो किसके खिलाफ नहीं करेंगे? सेल्फ के खिलाफ, क्योंकि उनके पास कोई ऐसा कॉम्बिनेशन नहीं है जो सेल्फ के अगेंस्ट रिएक्ट करता हो, बिकॉज ये वही वाले हैं जिन्होंने सेल्फ के अगेंस्ट रिएक्ट नहीं किया था। वो जो बच गए हैं, ये वो वाले हैं। वो जो बच गए हैं वो आते हैं हमारे सिस्टम के अंदर। They are the ones who can detect you, they are the ones who can detect bacteria, they are the ones who might detect viruses, but they will never ever going to detect or react against our own antigens। इस तरह से सेल्फ और नॉन सेल्फ का डिफरेंस बनता है। अब अगर आप इसको डिसाइफर कर लें, अल्लाह ताला आपको मुश्किल बातें पूछ रहे हैं मेरे से आप, दैट इज द टाइम, अगर एक न्यू इम्यून सेल्स इज अंडर प्रोसेस, आपकी बॉडी उसको सारे सेल्फ एंटीजन से मिलाती है, वो उनमें से फूफी को देख के कुरी मारता है, यू नो, किसी एक को देख के वो गुस्सा करता है, रिएक्टिव होता है, एंड योर सिस्टम फेल टू किल दैट सेल। और आपका सिस्टम उसको मारने में, उसको मारना चाहिए ना फौरी तौर पर, योर सिस्टम फेल टू किल दैट सेल एट दैट टाइम। अगर आपका सिस्टम उसको उस वक्त नहीं मारता, क्या होगा? दैट सेल विल सर्वाइव इन योर इम्यून सिस्टम, एंड दैट सेल जब भी वो आपकी बॉडी में जाएगा, जहां पर भी वो आपकी बॉडी में जाएगा, जहां पर भी उसको पुप्पी नजर आएगी, यू नो, जहां पर भी उसको वो वाला एंटीजन नजर आएगा, दैट सेल विल स्टार्ट रिएक्टिंग विद दैट एंटीजन। आप कहेंगे ओ भाई ये तेरी फुफी है, आप कहेंगे जनाबे अली आपका अपना है, आपका अपना मेंबर है, प्लीज 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 डू नॉट रिएक्ट विद दैट थिंग। बट आप इसको खत्म करना चाहिए था एट द टाइम ऑफ इट्स, जब ये डेवलप हो रहा था। इसको बोलते हैं ऑटो इम्यूनिटी। दैट इज हाउ ऑटो इम्यूनिटी इज एक्चुअली क्रिएटेड। अल्लाह थोड़ा भला करे, औखियां गल्लां पूछ ले, ठीक है? आर यू गेटिंग माय पॉइंट? मिल गया उसका जवाब?
यस सर, जी सर मिल गया।
वेरी गुड, शाबाश जी। काशिफ आप कुछ पूछ रहे हैं? देन यू पीपल, आई विल गोइंग टू आस्क यू टू प्लीज राइट योर नेम एंड आईडी नाउ। जिन्होंने पहले भी लिखा है वो दोबारा लिख दें प्लीज। प्लीज राइट योर नेम एंड आईडी नाउ एंड इन द मीन वाइल काशिफ आप अपना सवाल पूछ सकते हैं।
यस सर, मैं पूछ रहा था अभी आपने वो स्किन ग्राफ्ट मेंशन किया था, तो रिसेंटली ये मैं देख रहा था कि बर्न विक्टिम्स जो होते हैं
हैं?
उनको बर्न विक्टिम्स जो होते हैं, जो बर्न स्किन होती है, तो उसको फिश की स्किन से किसी तरह वो ग्राफ्ट कर रहे होते हैं।
रिसेंटली नहीं है,
तो
बहुत पुरानी
वो
बात।
चले वो मैंने जब देखा था तो ये क्या है ये,
कुछ
उस
नहीं है ये,
तब
effective
नहीं
नहीं है।
इसका कोई
नहीं देखो,
ऐसे ही है
जब हम कोई नई चीज डिस्कवर करते हैं ना, इनोवेशन है ना, हम अलग अलग किस्म के ऑप्शंस ट्राई
यस
करते हैं। इस वक्त मैं समझा तो इलेक्ट्रॉनिक स्किन की बात करेगा जो 2 दिन पहले हुआ है BBC में।
नहीं
मैं
नहीं
समझा तो उसकी बात करेगा, उन्होंने बहुत पुरानी बात की है। तो इनोवेशन है ना, आप डिफरेंट किस्म के ऑप्शंस को ट्राई करते हैं, डिफरेंट किस्म की स्किन को ग्राफ्ट करने की कोशिश करते हैं कि जिसको हमारी बॉडी कम से कम रिजेक्ट करे, या अगर रिजेक्ट करती भी है तो हम उसको डील कर सकते हो। तो उनमें से एक ऑप्शन वो है। दिस इज नॉट अ वेरी वर्किंग वे, अभी तक वो किसी भी लेवल के ऊपर अप्रूव नहीं हुआ है। लेकिन लाइक इनोवेशन ऐसी होती है, शुरू में आप एक वियर्ड लगता है, बाद में क्या पता वो हमारी बॉडी से मैच
ट्रू
करते
ट्रू
हैं। लेकिन यू डोंट अंडरस्टैंड, यू कैंट एक्सेप्ट, यू कैंट एक्सेप्ट नहीं, यू कैंट एक्सेप्ट एक फिश का स्किन सेल जो है ना, उसको हमारा इम्यून सिस्टम बड़ा प्यार मोहब्बत से अकोमोडेट कर लेगा। उसके लिए वो बिल्कुल दूर की बात है। बट हु नोस, कोई जैसे कोई कुछ भी हो सकता है।